Scientists at leading research institutions have created groundbreaking nanocrystals that can measure minuscule forces inside living organisms, opening new possibilities for understanding biological processes. The research, published in Nature, demonstrates two distinct approaches using specialized light-emitting particles that respond to physical pressure. The first team, led by Fardian Melamed and colleagues, developed nanocrystals that produce dramatic bursts of light when stimulated. These crystals contain thulium ions embedded within sodium yttrium fluoride. When force is applied to these nanocrystals, they require more power to generate their characteristic light burst. By measuring these changes, researchers can detect forces ranging from hundreds of piconewtons. A piconewton is 1 trillionth of a newton, up to 10 micronewtons. The foundation of this breakthrough lies in the unique properties of lanthanide ions, which are rare earth elements known for their distinctive light-emitting characteristics. When these ions are carefully positioned within specific crystal structures, they create materials that respond to external stimuli in measurable ways. The photon avalanching phenomenon, utilized by Fardian Melamed's team, occurs when a small input of light triggers a cascade effect within the crystal. This process amplifies the initial signal, similar to how a small snowball rolling down a mountain can trigger an avalanche. The researchers discovered that applying physical force to these crystals changes the amount of energy needed to start this cascade. The team observed 3 distinct responses to force application. First, they noted avalanche shifting, where increased force required more power to initiate the light burst. Second, they documented mechanobrightening, where the force caused an increase in the intensity of emitted light. Finally, they observed mechanochroism, where the force altered the spectrum of light emitted by the crystals. In a parallel breakthrough, researchers Kassar and colleagues took a different approach. They created nanocrystals using erbium and ytterbium ions. When exposed to infrared light, these crystals emit both green and red light. The ratio between these colors shifts when force is applied to the crystals, creating a precise measurement tool. The team demonstrated this technology's practical applications by measuring the bite force of tiny Caenorhabditis elegans worms. They embedded their nanocrystals in microscopic polystyrene spheres similar in size to bacteria. As the worms consumed these spheres, the crystals inside measured the force exerted by the creature's muscle contractions during feeding. The polystyrene microspheres containing these nanocrystals passed through the digestive system of the worms, specifically through a structure called the grinder. This tooth-like component uses muscle contractions to break down food particles. As the microspheres encountered this grinding mechanism, the embedded nanocrystals reported real-time measurements of the forces at work. Using a fluorescence microscope to monitor changes in crystal emissions, researchers recorded consistent shifts in light patterns that corresponded with electrical signals triggering muscle contractions. These measurements indicated the worms exert approximately 10 micronewtons of force when biting, a finding that provides new insights into how these organisms process their food. Despite the groundbreaking nature of these discoveries, researchers faced several technical hurdles. Temperature variations can influence how the crystals emit light, and material defects within the crystals required careful consideration. The research teams developed methods to compensate for these effects, ensuring their force measurements remained accurate across different conditions. Kassar's team's approach of using ratios between different light emissions proved particularly effective, as this method naturally compensates for some environmental variations. The implications of this research extend far beyond studying microscopic worms. Medical researchers could potentially use these tools to study muscle function in different organisms, including humans. The technology might prove valuable in understanding heart muscle mechanics or evaluating the effectiveness of treatments for muscular disorders. Many biological functions rely on mechanical forces, from cell division to tissue development, and these new measurement tools could help reveal how these forces influence health and disease. Scientists continue to explore new variations of these force-sensing crystals. By combining different lanthanide ions and adjusting crystal structures, researchers aim to create sensors with even greater sensitivity and versatility. The technology's development over just 4 years demonstrates rapid progress in this field. From the initial discovery of photon avalanching in nanocrystals to their current application in biological systems, researchers have quickly advanced these tools toward practical use. Current efforts focus on improving the reliability and ease of use of these measurement techniques. The research teams employed sophisticated equipment and precise methodologies to achieve their results. The atomic force microscope used in Fardy and Melamid's study allowed for exact application of force to individual nanocrystals, while specialized optical equipment measured their response. Future developments might include sensors capable of measuring forces in 3 dimensions, or systems that can simultaneously monitor multiple types of mechanical interactions. These advances could provide even more detailed information about biological processes. The technology might also find applications beyond biological research, potentially benefiting industries requiring precise force measurements, from manufacturing to materials testing. The combination of innovative crystal engineering, precise measurement techniques, and practical biological applications demonstrates the power of modern scientific research to create new tools for understanding our world. As these technologies mature, they may become standard tools in biological research and medical diagnostics, advancing our understanding of life's fundamental processes.